Proteins are extremely important molecules that are needed to maintain life. Every cell in our body contains thousands of proteins, which are essential for its structure and function. For cells to work properly, there must be a healthy balance of unwanted proteins being broken down and new proteins being made from them. This recycling process is necessary to prevent damaged or surplus proteins from building up and becoming toxic to the cell. It relies mainly on structures known as proteasomes. Any unwanted proteins are tagged with a chain of proteins called ubiquitin. Any protein with a ubiquitin tail means that it's destined for the proteasome. Proteasomes are shaped like barrels. They open at one end to allow tagged proteins to enter, and at the other end to allow broken down pieces of protein to exit. Only tagged proteins are captured by proteasomes. Once captured, the protein unwinds and enters the proteasome. Inside, the protein is degraded. The broken down pieces are then released and recycled to make new proteins. Sometimes proteasomes can become overwhelmed when cells generate large amounts of unwanted proteins. When this happens, a different clearing system is used. Instead, tagged proteins aggregate into structures called agrosomes. These are cleared by other structures called lysosomes, which get rid of the proteins instead of recycling them. Cancer cells naturally make more proteins than normal cells because they grow quickly. To prevent proteins building up, they must rely on proteasomes to work properly. This is especially so for myeloma cells because they produce large amounts of paraprotein which must be cleared. Drugs that stop proteasomes from working are therefore a very useful way of killing myeloma cells. By allowing proteins to build up, they eventually become toxic, causing the myeloma cells to die. Drugs called proteasome inhibitors have been developed to work this way, and a number are now used to treat myeloma. Others are also being developed to target escape routes such as the agrosome.